Good afternoon, everyone. I think we get slowly started. It's already late in the afternoon in different parts of the world, depending where you're listening in. And I'd like to welcome all of you to the Emerging Leader Series at James Cook University in Singapore. And this is the fifth of the series this year at James Cook, with a particular focus on food innovations and health aspects. We have three different speakers today, all focusing on the same topic, and I hope you enjoy and also can participate in these conversations today. So a couple of housekeeping notes. So we will record this webinar, so also for others who are not able to actually listen to it live today, can actually listen to it afterwards, so it will be recorded. All attendees will be able to ask questions through the webinar. So the question and answer function at the end, ideally, we can ask and answer question, but you're able also during each of the different presenters, we can answer some of the questions. And then, of course, at the end, we will have a round table discussion. There will be also a satisfaction poll and survey at the end of the webinar. So you can leave constructive comments also there, how we can improve or what you liked and disliked at the presentation. And if you have any kind of technical issues, you can use the chat button to communicate with the event organizer, and they can help you to organize any potential problems you have. So with this, I'd like to introduce our first speaker, Dr. Wasamon Nutakul, and I'd like to give you a little biography about her. She is a SciTech specialist at the Good Food Institute, Asia Pacific in Singapore. And she will give you just not much more information what the Good Food Institute actually does and what the aims are. But about herself, she has over 8 years of experience in the field of food science and technology. And she spent 5 years in the food industry, through which she gained expertise in food proteins and their different applications. Before joining the GFA, she spent also 2 years in academia and industry, the interface developing new talents for the food industry in Thailand. Wasamon herself holds a PhD in food science from the University of Massachusetts, Amherst in the USA, and is now putting her years of experience towards fixing the world's most pressing crisis, an unsustainable global food system. Wasamon, so it's all over to you. Thank you.
Thank you, Andreas. Can everyone see my screen?
Yes.
Perfect. Okay. So thank you for the warm introduction. Hello, everyone. It's an honor to be here today. So for those of you who are not familiar with the Good Food Institute, we are the global nonprofit organization focused on future-proofing our global protein supply. As a global think tank, GFI advances open source alternative protein science and research, mobilize funding and talent, and empower partners across the food system to accelerate the sector. We channel our work through 3 functional programs of policy, corporate engagement, and SciTech, where we work with policymakers to help them meet their policy goals with alternative proteins. We work with company and investors to help them seize the scale market opportunities. In SciTech, the team of scientists, we work to build a robust research ecosystem, creating resource for open access information, and creating a talent pipeline to bolster innovation. As a nonprofit, we are fully funded by philanthropy, which allows us to deliver our service free of charge and publish an open access research and to benefit a range of stakeholders across the industry. In terms of alternative proteins goal, we focus on 3 pillars of alternative proteins, which are plant-based meat, fermentation-derived protein, and cultivated meat. For today's presentation, I'm going to focus on plant-based meat, but I like to highlight that the trends we see more and more are the hybridization of these 3 pillars into next generation alternative protein products. We do believe that these alternative methods of protein production will help us reduce the impact of traditional protein production. As you can see from this slide, the plant-based meat use less land, less water, and create lower greenhouse gas as compared to that of conventional protein production. When it comes to plant-based product, it defined as a food product that are produced directly from plants and other non-animal alternative ingredients. These next generation of plant-based products are designed to look, taste, and cook like conventional meat, while also offer complex carbohydrate and fiber together with protein. The protein content and functionality are extremely important to create these plant-based meat products. Why health and environment concern brings consumer into the plant-based food categories, taste and price are still a key factors influencing acceptance or continued consumption. The perceived taste and price are among the top reasons consumers either do not eat or will not try plant-based food. This suggests that plant-based food companies should keep in mind about the price of traditional meat in the same categories and also focus on creating high-quality plant-based products that closely mimic the sensory attributes of animal-based food. From the study, we found that the top taste barrier reported for plant-based meat are moisture, flavor, and texture. We will look into the principle and value chain of plant-based meat to understand more of how research and development has been done to address this issue. First, I'd like to talk about the two main types of proteins. These are the globular proteins and fibrous protein. The globular protein act as functional and structural protein. It has irregular amino acid sequence. They are more water soluble and have tightly packed spherical shape. They are more sensitive to environmental conditions like pH and temperature changes. Alternatively, the fibrous protein provides structure and constitute protective components such as the fibrous structure found in connective tissue and muscle fiber in animals. Fibrous protein have repetitive amino acid sequence and are mostly water insoluble. So most plant proteins are globular and most animal meat proteins are fibrous. As a result, the core goal of creating plant-based meat product is persuading these global proteins to develop the function and structures of fibrous protein. To accomplish this goal, there are many opportunities across the value chain. This picture of the plant-based meat value chain can give you an idea of different researches that are going on behind our plant-based meat product. We first look into crop development, where you select a good crops candidate. Make sure it has the desired trait you are looking for. In many cases, these crops will need to be optimized by different breeding techniques to be suitable for plant-based meat application. Once we have the crops, the key ingredient, which is the protein, will be extracted out. Here, the extraction process and post-extraction protein modification is crucial to gain the functionality that will help the protein perform well in plant-based meat production. With the protein key ingredients and other functional ingredients such as fat, stabilizer, color, and flavor, the formulation work can start. Here, the knowledge on ingredient process interaction is very important in this last step because we need to get the ideal texture, flavor, aroma for the plant-based meat. Since crops are where we start off, we have to have a robust criteria when it comes to identifying the right crops. The majority of plant-based meat right now is still using soy, and this chart demonstrates some property of soy, pea, and wheat protein. These include protein concentration in the crops, their digestibility score, or representing here as PDCAS, allergen risk, flavor. Because these proteins have been available through other processes, they have high commercial stage and functionality score as well as low cost. However, there are many considerations that should be taken as listed in this slide when we exploring plant protein sources. Novel source with lower allergen risk, better flavor, as well as better consumer perception are continually being developed. If you'd like to know more information about this, the GFI plant protein primer outline different plant protein processing and choice parameter. So it can be a good framework for you, and you can go to our website to get the information on this report. Beyond crops, the aquatic plants are also promising candidates. These species have some unique properties in addition to having a high protein content, which ranges from 9 to 25%. They have high omega-3 fatty acid content, and this is very important when you consider developing a plant-based seafood, because By nature, seafood contain omega-3 fatty acid, and these omega-3 such as DHA and EPA is quite hard to find in the plant source. In addition, these aquatic crops also have scalability potential. It can promote whole plant harvesting. It's used minimal land, and some even have a relevant coloration that we look for in cooking the meat, such as the red seaweed, that it changes the color from red to brown when cooked. In 2022, we have seen some exciting development in the key ingredients launches, as well as companies exploring into upcycling ingredients for plant-based meat. Showing here in this slide on your right is a typical oilseed fractionation process. Recently, modification to traditional process like this have been made to enhance plant protein functionality, reduce extraction costs, and improve environmental sustainability. Some of the good examples are the partnership between Benson Hill and ADM. So Benson Hill has developed ultra-high protein soybeans. With the partnership with ADM, they plan to scale this to a high commercial stage. This is very promising because with the ultra-high soybean, it requires less processing, while it's retain a better protein quality. Equinome also successfully achieved 75% protein concentration with its minimally processed pea. And this means the pea protein resulting from here will actually have better function. Some other company also exploring the side stream of sunflower oil production, such as Apro and Becron, and they managed to actually create sunflower protein isolate. Rocket also launched rice protein together with their famous pea protein portfolio. The company Leaf is working on extracting protein from plant leaves, which is the most abundant protein on earth. Last but not least, ADM is creating an upcycled canola protein from their non-GMO rapeseed. Another group of ingredient I'd like to mention is alternative fats. Since taste and mouthfeel are key to get consumer acceptance with alternative proteins, lipid ingredients play a big role as it brings mouthfeel and flavor to the product. The challenge with alternative fat, which based mostly from plant source, is their differences in melting temperature and melting behavior. This drastically changes how the product is cooked, how the consumer experience flavor release when eating, as well as the perception of juiciness. So we are looking for the alternative fat that can deliver those flavor, texture, cookability, while at the same time being affordable and environmentally responsible. We see advances in 2022 with field development in alternative fats, where Givaudan have launched their Prime Lock Plus that is an encapsulated coconut oil that deliver both flavor and better cookability. Lipid, which is the startup that secured $4 million funding to scale up their phytofat production. They plan to be able to produce 10 tons of their product per year. This reduced more than 97% of saturated fat compared to just by using palm and coconut oil in the formulation. Good Food Technology is another company using cognac to mimic pork fat, and they also launched the consumer brand called Plant Seafood, which used their technology to create a plant-based pork dumplings. So we also see some activities in the academic realm. And so basically the common theme here is that plant lipids can be structured to improve their melting point range as well as cookability. So texturization is the next step that we need to look into after we got all the key ingredients. Basically, texturization denatures and analyzes the protein. It forms a fiber that resembles the muscle tissue. It produces the end product in the form that can be granulated, shredded, and chunks. Looking deeper into one of the most widely used texturization technology, which is extrusion. Extrusion transforms biopolymer ingredients like proteins and polysaccharide into semi-solid. To begin with, on your right, dry polymer powder are fed into the barrel that contains a twin screw. These screws used to push the batch forward. The ingredients are then mixed, heat, and undergo shear force the product are formed during the exit die where it gets cooled down as well as stretched out to form those fibrous structure. Most key equipment manufacturers provide twin screw extruders for high moisture extrusion and low moisture extrusion. The main difference between these two types of extrudate is the moisture concentration, where the low moisture you have lower than 35% and the high moisture you have higher than 50%. With the low moisture, the product will be right away dry after the extrusion. So therefore, these type of TBP are used more in the restructured product like a burger patty or sausages, versus with the high moisture extrudate, it is more suitable to be used to mimic a whole muscle cut. In this year, we see an advancement in extrusion where the two dies are introduced into the extrusion machine. Dr. Martin Hoffman from ETS Zurich have built this extruder that contains two extrusion die, one for the pea protein and the other one for the fat. These product then feed into one extrusion attachment that combine the protein and the fat into this marble-looking plant-based meat cut that replicates the appearance and mouthfeel of beef itself. In the last few minutes, I'm going to move on to other alternative texturizing technology. From this picture, in comparison with the process of extrusion on the top of this illustration, shear cell technology is showcased in the bottom. So shear cell technology also have the mixing and hydrating stage, which is done outside the shear cell unit. Then the product is introduced to the unit, thermomechanical stage where the shear force and high temperature are applied to the batch, and then followed by the cooling stage. The result product have the same desirable fibrous texture. However, with the shear cell, you can get a thicker slice of the texturized protein. Currently, researchers are working on scaling up this technology for a commercial scale. In the other technology on 3D printing, it has been utilized with so many companies such as Alfarm produce a 3D printed pea, soy, and algae-based Wagyu beef. Plantage from Israel printed whole-cut salmon fillet from legumes and protein and algae extract. Nova Meat have created a hybrid product. They do a 3D print on cultivated animal cells, combine that with plant-based derivative, fungi, algae, and spirulina. The last technology that I like to mention is the spinning tech. So the company called Tender is perfecting its rotary jet spinning technology to create a hair with fiber to organize and create a fibrous muscle tissue. So they actually developed this technology after getting a grant to develop cultivated meat scaffolding. So this one, I think they aim to work with the restaurant and formally launched in 2023. With the multiple part of plant-based meat value chain, more and more data will be gathered. Data collection and analysis will become an integral part of improving crops' genetic strategies, understanding the relationship between protein sequences, structure, and functionality, and comprehending consumer preference. Machine learning can be applied to use and collect data, analyze data, and predict a best working formulation. This can expedite the process and shorten the development time. So without that, I'd like to wrap up my presentation, and thank you all for your attention.
Thank you very much, Wasamon. This was a really nice, interesting, and very comprehensive presentation about this field of alternative proteins and plants. And I have already 2 questions for you, if you don't mind to answer them. We can have more questions at the end. The one question is from Jeff. He is a little bit concerned about the impact of transport. Also should be added to the impact to the planet. So, for example, pea proteins harvested in Canada, dried, shipped to USA, processed, and then shipped worldwide can be not a viable solution for the future. And therefore, the components themselves are all deemed good. But do you think that might impact, and if how?
Thank you, Jeffrey, for the question. So we do aware that this is happening, especially in Asia. A lot of manufacturers are actually import the products. So one of the project that working on in GFI is we look into crop diversification. So we start looking into local crops that is available here for manufacturing here. So in the sense of the transport load, I would say from the study, the transport right now is still lower than the consumption of beef itself in the production of beef itself, if you were to compare by ratio. But we are aware that this is not an efficient system. And so that's why we try to addressing this by looking into local crops, what is available, and connecting all the players that can make those local crops become a commercial commodity.
Oh, thanks very much for this, Wasamon. That's interesting to know. Second question from Denise. It's about the complex processes for converting plants into meat substitute. Is this really a reasonable solution, she asked, in terms of efficiency? Should there be, or are there a move towards getting people to accept plant foods in a less processed state?
So the way we see plant-based meat product is almost like introductory product into more of a plant-based lifestyle, because plant-based meat itself is designed for, say, flexitarian and general public. So for them to right away convert to a full whole food plant-based meat is going to be quite difficult. So the way we see this is a great tool to introduce people to a more plant-based lifestyle right now, and basically lowering the hurdle for them.
Yeah, that's absolutely right. I mean, from our own experience, my wife is vegetarian, and she's not interested in eating something which tastes like meat. That's a fact, and that's quite reasonable to understand this. There was actually a third question coming just in. Will alternative protein foods contain also vitamin B12? That's a very specific question.
So I believe many companies will try to match, because B12 is actually lacking for a lot of vegan and vegetarian items. So B12 is one of the target micronutrients that will be fortified into alternative protein products.
Okay, makes them more healthy, more complex, more comprehensive.
More complete. Yeah,
more complete, right? Okay, thanks very much for this and the question, and we will have some more questions to answer perhaps at the end when we have a more kind of panel discussion. Thanks very much, Wasamon.
Thank you.
So with this, I will introduce you to the next speaker, which is Mr. Mihir Pershad. Mihir is CEO of Umami Meats. It's a company based here in Singapore. And this is a pioneering company, basically, a startup cultivating the future of sustainable and not caught seafood, which is very different from most of the plant-based food we heard just now. As a CEO and founder, Mihir is building a team developing delicious, affordable, sustainable, cultivated, and basically cell-based fish that is better for our health, our oceans, and our future. He believes that nature is our biggest teacher and is dedicated to preserve its delicate balance by thinking differently, honoring tradition, and protecting the taste of the sea. Sorry, Mihir's academic background is in protein biochemistry, and he has spent his career working to commercialize deep tech innovations to address humanity's great challenges. Most recently, he was a venture partner at Early Charm Ventures, where he led the commercialization of university spin-outs at the intersection of biotechnology and aquaculture. Mihir is also an author of a book called Cultivated Abundance, which outlines a framework for entrepreneurs to leverage transformative technologies to build an inspiring, abundant future for humanities. And that's really a very interesting book to read if you want to take it further and not just listen to what we say today, but also have your own impact on this. All right, thanks very much, Mihir. I'll leave it all to you now. Thanks.
Thanks very much for the very, very kind introduction. I'll pull my slides up here and then I'll get started. Are you able to see my slides all right?
Yes. Very nice. Thanks.
So, yeah. So thanks everyone for joining today. I'm looking forward to telling you a bit more about what we do at Umami Meats and more broadly about the cultivated seafood opportunity to build more complementary products that, like the plant-based, can help to shift demand from depleting the oceans to potentially more sustainable solutions for the future. So the reason that we are focused on seafood is that the supply is under a grave threat from a number of major factors and forces. Number one is that we want to eat more seafood than is currently available via wild catch. And then for the last 50 years, we've been supplementing that with farming. But farming is still a limited production method in many ways. And we're only really able to produce a handful of species, about a half dozen or so major commodities that are produced via farming. And the result is that seafood supply has doubled globally, largely as a benefit from our ability to farm. But there are still over 24 species that are $1 billion in sales a year that are being pulled out of the ocean at unsustainable levels. And this has led to 93% of fisheries being considered to be fully fished or overfished. And fully fished is a bit of a misnomer in a lot of ways, because we allow fisheries to drop to 10, 15, 20% of historical levels before we start to consider them to be under major unsustainable threat. So we basically get pretty far down the path toward really driving species to the brink of extinction before we start to consider them to be unsustainable. So I think actually reevaluating some of our early definitions here is a critical part of making sure that we are really producing sustainably. And compounding and worsening the challenges here in seafood production is that climate change is making the food system less predictable. Particularly when you come to the oceans are sequestering almost 2/3 of the carbon we put into the air every year. And this is leading to rapid acidification, as well as rapid temperature changes, as water holds energy quite well. The negative kind of side effects are that we see warming in unpredictable ways in the oceans because of the way the currents help to move and distribute heat. And so some fisheries are being impacted much more rapidly than others. But overall, the general trend is toward fisheries not being useful as a source of food as early as mid-century, but certainly by the end of the century, if we don't change what we're doing. And then we add on top of that the increasing demand as more and more people come into the middle class in highly populous countries. And as they come into the middle class and have increasing purchasing power, they also have an increasing desire to eat animal protein. This is something that we've seen in a dozen countries over a few decades. It seems to be quite a consistent trend. And the key challenge is we have to find ways to produce additional protein from the oceans and not just replace our current production levels. Asia is going to represent 70% of seafood consumption by the end of this decade, and we're already somewhere in the 63 to 65% range for global demand. But broadly, not just Asian consumption is increasing, global consumption is expected to be 78% higher than 2020 levels. And this is something that we have to develop novel technologies for, because the demand that's rising is often for unfarmed and unfarmable species using current technology. And while some of these species could be farmed in a reasonably sustainable way, given enough time, we don't really have that time in front of us. If you take the example of tuna, for example, it took a couple of the big keiretsus in Japan investing tens of millions plus over 35 to 40 years to successfully enable the biology for farming of bluefin tuna. And that's been done now. As of a couple of years ago, we are now able to farm bluefin tuna. The problem is that we still cannot do so economically. And often, currently, I believe the system is quite far from a true economical replacement for wild-caught tuna. And so we need solutions that can be implemented on a shorter time scale and that can scale quite rapidly, given the development time still ahead of us. So the question that we landed on thinking about how we could solve this problem is that fundamentally we have to figure out where we're going to get more fish. And going through this thought process, we started thinking that actually when you raise a fish, there's a substantial portion of the animal that you rear that you catch that is not eaten by humans consistently. So this is typically 40 to 50% of the fish by weight, depending on the animal. And so we started thinking that potentially if we could produce the fish without having to grow the whole fish that includes the inedible or uneaten portions, that we might be able to create a more sustainable food system without having to produce quite a lot of uneaten biomass that would then be used in not efficient ways. And at Umami Meats, our solution to this is crafting sustainable seafood that's cultivated, not caught. And cultivated means a variety of different terms that you may have heard. It could be cell-based, you may have heard lab-grown, you may have heard cultured. Cultivated is the term that we use to describe this process. And this process entails starting by taking a small tissue sample from a fish, usually a couple of grams, about 1 to 2 grams on average for our process. And then we take that very small sample and use it to isolate a stem cell line. And in our case, we're isolating stem cells that produce muscle and fat and connective tissue, all from a single stem cell source. These are called mesenchymal stem cells, or MSCs. If you've worked in human stem cells, you've probably come across them. They tend to be quite used frequently in human therapeutic work. So we've taken that and are now the first company to be able to establish mesenchymal stem cells from fish. We isolate those stem cells and then produce a working stock in a cryo freezer. And then as we need to go into production, we then take one of those vials out, put them into a bioreactor and culture from just about 1 million or 2 million cells into many trillions of cells. And trillions of cells sounds like quite a lot of cells to grow. But in fact, a kilo of meat is about 1 trillion cells, give or take a few million here and there. So actually, we need to produce tens and thousands of trillions of cells to produce large volumes of seafood. We then use a proprietary process we've developed to turn these stem cells into mature muscle and fat in just a few days, actually still in the stirred tank bioreactor. And then we collect out that mature muscle and fat and structure it into a variety of seafood products. We work on both hybrid products, which would be fish, muscle, and fat incorporated into a plant-based with an ingredient and then structure, as well as products that are going to be 100% cultivated. Those will take longer to market though, as that technology is less mature for structuring. One of the things that we think is really exciting about cell-based foods that are cultivated is that cultivated fish, for example, can be more versatile in application and in targeting of consumer desire than traditional fish. So in traditional fish, you're often looking to grade the fish in the case of sushi and surimi and sashimi, for example, whether you get something that's high grade, that is sushi and sashimi grade, or whether it's small pieces that are too small for a portion that maybe get pooled and eventually turned into surimi or a fish paste. The ability to produce hybrid plant and cell-based seafoods enables potential to create a variety of products that are much more difficult to produce from fish, or that are seasonally limited, or where you only get 10% of the fish with the high grade cut and 80% with a moderate or low grade cut. And so there's a real opportunity to create high grade fillets and steaks, make sashimi more available and more affordable using cultivation. And beyond this sort of versatility, the thing that we think is quite interesting is the ability to produce fish that's better for our health and better for our oceans. So when I talk about better for our health, we look at a couple of things. So one is reliability. The best availability of food, or the best food is that one which is available and consistently so that it doesn't create instability in the supply chain for people. And in our view, we can also produce this fish that's free from mercury, antibiotics, microplastics, and parasites. And this is something increasingly of concern to consumers, especially if they're purchasing fish like tuna that has been known for decades to be potential concern to some consumers, depending on particularly young children or pregnant women. We can also help to decrease greenhouse gas emissions. We have estimates of this right now, but until the products go into a full life cycle analysis using our process, we don't have exact numbers. But actually, potentially the largest impact to the comment earlier about supply chain is that we can locate production of fresh seafood adjacent to where consumers are. So we can put production facilities outside of Seoul and Singapore and Tokyo, and make sure that seafood is not having to travel thousands and thousands of kilometers in refrigerated air freight to make it fresh to consumers. we can also help to protect and restore biodiversity in the oceans. The oceans are incredibly resilient, but we have to give them time and opportunity to recover. And the same goes for the species that we are consistently overfishing. And lastly, between 25 and 30% of fish comes from plastic in the oceans comes from fishing fleets. And so the ability to reduce the amount of fishing fleet required to supply us with fresh fish means that we can also help to reduce one of the key point sources for plastic ending up in the oceans. But let's say that we're all bought in on the idea of producing some species of fish via cultivation. I think the key question then, which is relatively unique to seafood, is what species do you work on? We don't have just one or two species of cow or one species of sheep to work with. We have dozens. And so as Umami meets, we decided on the following criteria and framework for determining what to focus on. And the species that we work with are those that are hard to domesticate. And we say this difficult to domesticate because either they cannot be farmed, or in the case of like a bluefin tuna, they can be farmed, but not economically. These are also then species that are on the IUCN red list, and we look a little bit adjacent to the red list as well for species that are considered vulnerable. But these are functionally species that are in serious decline and that are facing threat of extinction if current trends continue. And then finally, we look at strong consumer demand. And so what do consumers want to continue eating more of? And if we combine these three criteria, you almost always land on a fish that's also pretty expensive because more people want to eat it and the supply is diminishing. And the premium price is more of a starting point for us, but our thinking is that we have to launch production and get it up to scale. And so inherently early stage production will be slightly more expensive, and we'll be able to reduce the cost with time and target lower and lower price species over time. We've chosen to start with Japanese eel, big-eyed tuna, and red snapper as the first species that we're going to target. But beyond that, we'll then be working on trying to expand that in the coming years. And as I mentioned, we have to start at a relatively higher price point because the technology is not at the point where we can compete with tilapia and carp today. But the strategy is to go from something that is more expensive and focus on species that are at price parity. So in a situation where we can manufacture at pilot scale, for example, we can manufacture in the ballpark of an $80 per kilogram fillet, which is on par with something like crab or eel. And in this case, one of the reasons we're focusing on eel is the higher price point. As we get into a full-scale commercial production, we'll be moving more toward a $25 to $30 per kilo price point, which is still relatively premium, but starting to get towards species like grouper and a broader range of fish, and actually pretty close to the price of salmon in Singapore, for example. And then as we optimize this process over the next 3 to 4 years, our target is to really hit production at $15 and below, which makes most seafood pretty close to price parity for consumers. And the opportunity in cell-based and cultivated seafood is quite substantial. Seafood is about a $215 billion market based on current growth rates to 2030. And cell-based seafood, if we can produce a cultivated product that matches on taste and texture, can take about 10% of that market by 2030 and potentially grow beyond that. And so we look at that and say, in that range, if we look at just the cultivated eel, red snapper, and tuna, there's about a $4.5 to $5 billion opportunity by the end of the decade to produce cultivated protein just for these couple of fish in a variety of formats. And that means there's tremendous opportunity for more players in the space and for more people to work on different species that we can't target today. As I mentioned, there's something in the neighborhood of 24 species that are consumed at levels that are putting wild-caught fish at risk. And so we would love to see others come into this space, either to work with us to expand how many species we can work on, or to work on developing additional solutions that can compete in the market for helping consumers to adapt to cultivated technology. And the last question that you have to ask, thinking about how do we make this actually reach this kind of scale, is what kind of business model is needed to maximize impact? And our view is that rather than trying to build an individual brand and have to grow everything ourselves, we want to be a B2B tech enabler, a true Intel inside concept for the seafood market, and to work with the world's biggest seafood and food processing companies to manufacture cultivated seafood for a variety of consumers, a variety of markets, under licensing arrangements and joint ventures, where we can basically enable and power this revolution into cultivated fish, while making sure that we can scale much more quickly than we could by ourselves. And just to give you a sense of how we're working in the market right now, so we actually did a first product demo just about 2 weeks ago with a fishball laksa that we showcased to a room full of VIPs in Singapore as part of the Singapore Restaurant Week. We're now working toward a battered fish fillet that's going to be launched in fish tacos at the end of October, as well as a grilled eel product that will be demonstrated in January of next year. And our longer term strategy is to move toward this very high end of the market with sushi and sashimi grade, and to start making that more affordable, as well as then thick cuts like steaks and thick fillets of fish like halibut that we think will take longer to achieve the right properties to get consumers on board. But I think cultivated is an exciting way to enable consumers to eat the fish they want while still having fish in the ocean for future generations. Looking forward to taking your questions. Thanks so much for your time.
Thanks very much, Mihir. This was a very nice, all the different talks about seafood, and at the end, the dishes you actually present makes me really hungry already at this time of the day. And it looks very appetizing, so really very interesting. So we don't have direct questions to you. I'm just going through. Here we have a question from Jeffrey. Mihir, can you touch on what technology hurdles have to be overcome to get the cultivated fish costs down per the charters you have showed? Many show that the cost reduction, but reality often hits the speed of the product reduction is too slow to fit the funding models. So yes, what do you think about this? Yeah,
that's definitely true, and it's an astute observation. I think the nature of a venture funded company is that you have to make big promises on tight timelines, and often those timelines get missed. I think our strategy is starting from the fundamentals. So we've gone and recruited some of the world's most experienced fish cell biologists to understand how far we can push the limits in terms of identifying the right fish cell types. One of the core things, if you think about a production process involving cells, most of your cost reduction is fairly linear. So you can reduce the cost of media, replacing some pharma grade ingredients with food grade. That's a pretty standard practice at the moment. You can also try to optimize maybe the hardware of the process to make it more efficient. We don't like optimizing the hardware as much because it, as to your point, lengthens the timeline of development and scale. So we focus more on process optimization, things like growing more cells at higher density, because then you can use the same hardware more efficiently. But actually the biggest lever in the production process is how fast your cells double and how many cells you can grow in a certain space, because it's the only exponential function you actually can control. So the cells are actually growing exponentially. And so if you can get them to grow another 1 or 2 doublings, you end up with 4 times the biomass in just an extra 2 days, for example. But if your cells double every 2 days instead of every day, everything gets drawn out much, much longer in your production process. And that has a massive impact on cost. So our view is get the cells that double the fastest and try to grow them to the highest possible density. And those are the biggest levers that we think we can pull to rapidly drive costs down.
Makes sense. Yeah. There's another question from Chris. If you can say something about the nutrition levels in general terms, provenance, energy intensity, and so on about these cultured fish.
Yeah. So I think there's a couple of ways to think about this. One, so provenance, we start actually working with traditional farms and with suppliers where we can trace lineage all the way back to the fish. So for at least from the cell point of view, we're getting something from the fish that's ideally the closest to what consumers would chase when hunting for a product. Nutrients right now, if you look at a pharma grade medium, those are typically soy derived amino acids, corn derived sugars, that they're coming from your typical commodity crops because those are the cheapest base ingredients and the most reliable for sourcing. One of the things that we're doing is actively trying to replace, for example, soy derived amino acids with algae and kelp derived amino acids and hydrolysates, because they're much better for the environment in terms of not needing industrial fertilizers and these sorts of things put onto the land, but they're also potentially carbon negative depending on what's done with the byproducts from that harvest. Energy intensity is a tough question to answer kind of in the abstract, but I'll say the way that we think about this is we're growing cells that don't have to spend nearly as much energy on homeostasis than an animal does, because they don't live for 2 to 4 years before they're harvested. So in terms of energy, we're getting much closer to plant trophic levels than you are to, you know, one level above that, because we're taking a plant or an algae input and feeding cells directly.
Yeah, and that's what all happens basically in the bioreactors, which you mentioned earlier on. And there comes a question from Jeffrey also. The independent of the speed, when these cells are doubling, there seems to be an assumption in the model that the cell quality is independent of the doubling time. Or could it be that the cells are reducing quality or nutrition levels?
I think what we see is there's a lot of genetic diversity even within a single organism. And between fish are quite, we call it genetically plastic, right? So you see a lot of genodiversity amongst populations that live together and that share a common parentage. So one of the benefits of working with fish is that if you sample from 10 or 12 different fish, you'll find that some of them grow much faster, some of them grow much slower. And you see this reflected in fish farming as well, where fish have to be graded routinely because they grow at very different rates. So our view is that seeing that at the macro level actually translates to the micro level, and that within the cells, there are some cells and some cell types that will grow much faster and some cell types that will grow much slower. For example, in our hands, we find that muscle stem cells tend to be quite slow growing relative to, example, mesenchymal stem cells, which are much more rapidly proliferative in the abstract. So if we just say this at a high level. Yeah.
Okay. Yeah, thanks very much, Mihir. I think at the end you might have some more questions to everybody, but we're going now to the next speaker, which is Dr. Thimo Ruethers. Dr. Ruethers is Research Fellow in the Human Health and Aging pillar within James Cook University here in Singapore. And the Tropical Futures Institute, one second, please. The Tropical Futures Institute at JCU is their primary research activities on molecular interaction between food and humans, with an emphasis on the immune system. The aquaculture products novel foods are of particular interest to Dr. Ruethers, particularly emerging from the ambitious Singapore food story goals. Thimo is a researcher and science communicator who has been tackling food allergies and promoting food safety worldwide. And he was based not only in Australia, but also in Austria, Germany, Philippines, Thailand, and Vietnam. He now focuses on Singapore's healthy, safe, and sustainable food future, utilizing over a decade of international interdisciplinary research experience. Thimo holds a PhD in medical molecular sciences from JCU, and both a bachelor and an MSc from Heinrich Heine University, Düsseldorf, which is in Germany. He seeks to develop to overcome challenges in securing humanity's alternative protein needs, which we heard earlier on, under the careful consideration of food safety, allergic diseases, and planetary health. Thimo, you have the floor.
Thank you very much, Andreas, for the very comprehensive and kind introduction. And also many thanks to Wasamon and Mihir to be here today and taking us for a really fantastic dive into the world of plant-based and cell-cultivated meat. And as Andreas briefly introduced, it is now my pleasure, I would call it, to take you for a walk on the allergy tightrope and some novel food challenges and opportunities. And as you heard earlier already, we are facing various major challenges requiring global action. And for that reason, the United Nations member states adapted 17 Sustainable Development Goals with a highly sustainable, highly ambitious agenda until 2030. And as we all know, food is linked to all these goals. And both food security and sustainability are key to reach them. And of course, we do need enough food to end hunger. The food must be safe, and it must promote good health and well-being. And it must be produced, distributed, and consumed responsibly and in a sustainable manner. And in particular in Singapore, we are committed to basically, I have to change this, yeah. Especially in Singapore, we are committed to a sustainable future, and we've implemented this global agenda on its own food story. And the plan is to really diversify the imports while growing both local and overseas. And this is really the key to the success. As we know, Singapore depends on the success of this story. And we have those three baskets. So it's about the diversification of import sources, growing local and growing overseas. Let me give you some examples beyond what Mihir and Wasamon were talking. Just this morning, actually, I read that now researchers at NTU managed to grow vegetables using human hair instead of soy. So that just came out today. And here on my left or right, I'm unsure about that, we have a colleague at NUS. He is basically just looking at this variety of kale and many other vegetables and try to tailor them for the indoor farming. He basically got a $10 million investment to create a research center. On my other side, we have my colleague Jose Dominguez from James Cook University. He's very much into the urban aquaculture of fish. And just above him, we have the hand of Professor Dean Jerry, who is really working on the next generation of farming shrimp prawns and making it tastier, better, more sustainable, and in particular, healthier. And for example, just talking about fish again, the Straits Times just in January 22 this year, they had a headline, new high-tech trout farm to boost Singapore's annual farm fish output by about 25%. So there's a lot happening. And of course, Singapore's international reputation, geographical location, growing population, and the limited resources puts everything from pre-production to post-ingestion basically under growing scrutiny. And especially if we look at the second basket of growing local, this is where we really have to tap in. And this is where we have to empower Singapore's industry to produce these, it says on the top right, 30 by 30, to produce 30% of nutritional needs by 2030. And at the moment, we are below 10%, if in case you're not aware of that. And this is obviously difficult. Space is very limited. Currently, we are utilizing about 1% of the land for the food production. And obviously, that's where from farm to table is not sufficient. We are trying to move from high-tech farm to table, but at the end, we really have to move from lab-grade farms or just labs to basically chopsticks. And major problems we are facing there is species adulteration and fraud, because you sometimes do not know what is coming from the farm, if it's grown local or in particular overseas. But also a very critical question is, what is the difference between a wild fish, a farm fish, a labbed fish? That's a part we are already looking at with some example species, red snapper and barramundi, looking at just what do we find in a barramundi wild caught, a barramundi farmed in urban aquaculture, or simply produced by Umami. And in this case with Umami, it's obviously the red snapper, as we did here before. But there are many other examples. Here we can see that we have some forgotten, really, sources, algae, microalgae. And then here, on the other hand, we have insects. Insects in some cultures have been a staple diet for many people worldwide, and they seem to be forgotten. And the United Nations reckons that this will be really the future, how we can feed the world. Saying sustainable source of protein for food and feed, that's insects. That's where we have to invest in. But then, of course, we also have the other parts. We heard about cell-cultivated fish. We also have cell-cultivated chicken. And then there are also cell-cultivated milk and many other products. And when we always talk about something new, and this comes with the Singapore food stories, there are always opportunities, risks, and challenges. And altogether, we can say that novel foods will definitely bring novel benefits, but also novel risks. And therefore, we have to really adjust our dietary and food safety strategies. And this is exactly the part I'd like to elaborate now a little bit further. But just before that, I just want to redefine again what was said earlier, but just very briefly. So alternative proteins, we have the plant-based. Wasamon was talking a lot about that. Mihir was talking about the cultivated. We have the fermentation. And then we also have many, many others I just mentioned earlier, novel foods. And if we go to a supermarket, this is really what I found. Basically 2 freezers or fridges full of plant-based meats, not really anything about fermentation. And now just above me, this is the only cell cultivated meat available in the market in the world. It's supposed to be chicken. The one who tried can make their own opinion about the taste, the texture, and everything around. And then we have, of course, the cell cultivated fish, the cell cultivated also shellfish, geoc meats. I will come to that a little bit later. So this is already basically just visualizing on one brief look. At the moment, plant-based food is leading the way forward, but others are catching up. Now, when we think about plant-based meat, Wasamon was touching it on very briefly, we can see that there's an allergen risk, that this is a major, major problem with plant-based meat. Now, let me elaborate very briefly on that further. Here we have birch pollen. Patients who are, or people who are allergic to birch pollen have a major, major risk to many, many other foods. And you can see here soy and many other ingredients for plant-based food. And there's this well-known cross-reactivity among food and pollen allergen sources. And I'm, for example, allergic to birch pollen. And one day I tried when I was overseas, so I was growing up in Germany. You obviously don't get jackfruit, what's here on that slide. Some of you may recognize it. And that was the very first time I was exposed to it, and I was highly allergic to it. The reason there, it's an allergy to birch pollen. Another cross-reactivity, that's how we call it, is then between latex and kiwi. And this just highlights, it's not even about the allergen to the actual source. It's also really about cross-reactivity to some ingredients or some inhalant allergen sources you would not even be thinking of. And what we've seen earlier, I just go one back. All this, what we here see nice with dots, red, orange, green, is not really known for the cell-cultivated meat. And cell-cultivated meat, Mihir touched on that briefly, it's the opportunity to really avoid adverse reactions to seafood. We can't have scrambled poisoning, really. We can't have ciguatera-toxin. We can't have marine biotoxin. We don't get parasites in that fish. And this is fantastic. That's touching the bit of toxicology. And this is where really the cell-cultivated meat is having a big, big bonus. We don't have heavy metal and all the other bits. On the other hand, though, one risk does remain, and that's this one here. The risk of immunological adverse reaction of allergies. Here, this is obviously an example from seafood, but this applies to all sorts of foods. Now, since I talk about allergies, let me just give you a very brief background. What are allergies? Allergens, we basically have a harmless protein. Upon recognition by immune cells, we have IgE antibodies produced. They are then presented on immune cells. Upon subsequent exposure to the same or similar protein, a whole cascade of immunological reaction is triggered, and we do get allergic reactions such as anaphylaxis. Now, sorry. This is fairly simple, but it gets more complicated. Here we have a fish, basa, a catfish, but there's obviously much more than just this one protein. That's the major allergen parvalbumin, but there's much more. Here we see the 5 previously very well-known allergens, and recently these have been more than doubled. So we have a quite high diversity of allergens, and they're all different in every species and very different from one and another. And this results in what we call a limited or a very high cross-reactivity in some cases. And that brings me to the challenges of managing allergies, or as an example, fish allergies. So if we have a fish, I mentioned antibodies recognize the protein in the allergen, we get an allergic reaction. Now the question is, what happens if we have another fish? We may or may not get an allergic reaction. And that's the challenge to figure out. And that's part of our research really we are doing. And I'm talking here about fish because fish and shellfish all together is combined as seafood and is a major trigger of allergic reactions worldwide. The allergy is often serious, rapid, lasts a lifetime. And unfortunately, we have insufficient diagnostic and management tools. And altogether, often the best a doctor or individual suffering from it can do is avoiding all seafood altogether. And this is not just the case here in Singapore or in the Asia Pacific. This is really a global thing. Globally, we have over 500 million people suffering from a food allergy, and there's a true rise of allergy and anaphylaxis. And seafood is indeed the predominant cause of anaphylaxis worldwide. Here, we unfortunately do not have data for Singapore, but we know that also here in Singapore and the whole region, seafood is the number one trigger of anaphylaxis. And even seafood gets much more shellfish in particular gets much more complicated. So we have crustaceans and mollusks, and then we still have cross-reactivity with mites, nematodes, and insects, edible insects, inhalant insects. Highly complex, and this is also where the food safety aspect comes in. But there's obviously more than just tropomyosin and arginine kinase here mentioned on the size. I did lose a little bit the track of time, but I think I have to move on a little bit faster. Apologies for that. We talked here about different allergens, and this is just probably a good overview to look at what is there out there for seafood. So we have fish, many different fish, bony fish, cartilaginous fish, related other vertebrates. And if we do a look at allergy assessments, we also have to take into account cell-based or cell-cultivated meats, and the same goes then for crustaceans. Here it's now on top of me, but it really belongs more to the crustacean, because to date, there's no company, to my knowledge, working on cell-cultivated mollusks. And let me almost wrap it up. So when we look at the allergenicity of novel protein sources, we have in silico predictions where we can use tools co-developed with A-Star named Talacat Pro. We have in vitro evaluations, and we can utilize clinical data or assessments to really predict the risk there is for the allergic reaction. And in addition, we can also then have biosensors to detect the allergens, or to even work on hypoallergenic traits, production, processing, or other bits. And altogether, we really have to look there at safety assessment and risk predictions on both ends. How it looks then in reality, here's just an example. This is an example where we can have in silico predictions utilizing genomic transcriptomics and looking at both sequence and structure, where we can then look at what are the different allergens from many different sources, here with the example of a catfish and a prawn. And there's more to it, even if we do detect and understand the presence or absence of allergens, these allergens can still be processed. So this goes then into the life cycle of allergens, where they're usually intact at the very beginning, but they then degrade over time, or they can be altered, which does then impact the allergenicity. And talking again about seafood and going back a little bit to what Mihir was talking earlier, that we are at the point where we need more aquaculture because there is no more wild fish. And then that's where we also have to boost our alternative seafood. The alternative seafood, I believe, we can control, we can amend. The aquaculture we have to investigate, and it might be controllable to some extent. And you might have an idea, a feeling by what I presented just now, that what we know so far about fish and seafood allergy is really under-investigated, and that obviously reflects also on all novel foods. And with that, I'd like to finish. I hope you managed to follow me on this walk of the allergy tightrope. And we really try to work on food safety here at JCU and the Tropical Futures Institute, looking at toxicity, detection of toxins, detection of allergens. What is the best way to label? How do we improve allergy diagnostics? How can we evaluate the allergenicity, prediction, alteration? And we try doing all this basically through translational research. We could say we have the vision to enable safe, healthy, and informed dietary decisions, and in particular with a view on the sustainable development goals above me. Thank you.
Thanks very much, Thimo. This was very comprehensive. Thanks very much for this. And we have a few questions here to Thimo and the rest of the panel. The first answer is, there's a question or comment from Geraldine, that the TFI, the Tropical Futures Institute and JCU Singapore, should visit the inaugural Agri-Food Tech Asia trade show in October, and that the Austrade is a big supporter of this event. Just to answer this, yes, James Cook University Singapore actually has a stand there themselves and will present some of our research, particularly in aquaculture and also on the food safety aspect. So yes, we will be there. A question from Jeffrey to you, Thimo. Is there a simple explanation why so many suffer from seafood allergy, but perhaps chicken allergy is almost unheard of?
Well, I wish there would be a simple explanation, but if there would be one, we would probably not be here. I can't give you an answer to that, unfortunately. So indeed, chicken allergy is almost unheard of. And interestingly, chicken allergy is sometimes linked with fish allergy, because the allergens I was talking about earlier, they are similar in both chicken and fish. But this is really, really rarely ever heard of. We know that for seafood allergy, that the prevalence of the allergy is often correlating with the abundance, with the consumption in a specific area. But another approach, and maybe Andreas wants to elaborate on that later, is that we know that crustaceans are very similar to mites. And in Singapore, for example, 7 to 80% of the population are sensitized to mites. And in mites, the allergens, including tropomyosin, the major allergen, is very similar to the one in prawns, crustaceans, and other shellfish. And it seems like there might be some cross-reactivity playing a role, but this is really something we are still investigating and hopefully know more one day soon, or 1 year soon.
Yeah, maybe I can add to this. This was a very good answer. I mean, when you look at house mite allergy, it's about 30% of the world population has allergic reaction to house mite. So the human body, the immune system, anything which is very foreign is easier recognized as foreign, and the immune response makes particular antibody to it and has an immune response to these foreign proteins. As close it is to us, as less likely we make antibodies. Otherwise, you have so-called autoimmune disease, which we know is also increasing. So chicken is a vertebrate and is much closer to us phylogenetically than a fish. And that is maybe one of the reasons they are closer to us. And there's very little beef allergy or mutton or the sheep allergy known, as well as chicken allergy. But as further we go away from the food sources, we know about many allergies, and probably plant allergy or pollen allergy is even much more prevalent than allergies to animals. So that's kind of might help Jeffrey with the answer to this.
Because even rarer than chicken allergy is a pork allergy.
There's another question, anonymous. Should we switch to becoming vegetarian rather than making artificial meat? I'll throw this in to everybody here, Wasamon and Mihir. What do you think?
I think that's similar to what I discussed before, that we will always gonna have consumer who have a hard time switching. And I think that's why I see that the role of like alternative protein product is there to kind of introduce them to a more plant-based or alternative protein lifestyle. Because it's actually not easy for people to switch, especially like, because I've had recently my friend just right away she switched to being vegan, and right now I have to keep reminding her to eat more tofu, or you need to study of what gonna be your protein source before you directly switch. Yeah.
Yeah, and of course, it has to be tasty. I mean, at the end of the day, you can do healthy food for an extended period, but if you want to do it for your lifetime, it's easier if you have a combination of taste and health. Yeah, Jeffrey was just thanking you for this question. Mihir, you want to add something to this?
I think that's a pretty comprehensive response. I mean, our view on cultivated as opposed to other proteins is simply that we have to make a very dramatic change in consumption patterns in 25 years. And food habits are some of the hardest to change because they're the most culturally driven compared to many. I mean, compared to buying different clothes or taking a different mode of transport to work, changing the food you eat is something that's very hard for people to do. Especially later in life, because it comes with all sorts of cultural implications that people are sometimes more tied to. So we think making that behavior change feel as small as possible is the most important way to make that happen.
And started early in life, basically. Also our new generations, not just of scientists, but also consumer, exposing them to this new world of food and our new world of living. Yeah.
Of course. And I think I don't take such a view that people will always eat meat. I think in 100, 200, 300 years, the conversation will look very different than the one we're having today, right? But I think we're talking about needing to make changes between mid-century and today, right? So that's the time horizon we look at for creating change right now.
Yes, I have a question from Guy. Is cultivated meat, fish, considered vegetarian, or are the cells enough to be considered flesh? That's a good ethical question.
I always say people have their own definitions, right? I think, but I would say we're using cells from an animal. It's meat, right? I think now whether veganism is a harder question because it depends why somebody and how somebody consider themselves vegan, right? If it's for animal cruelty reasons versus if it's just anything derived from an animal, then if you want something that doesn't harm animals, then maybe it's vegan by your definition. If you want something that wasn't from an animal, then it's still not vegan. So,
okay, another question earlier was also for you, Mihir, regarding the fish. When you take your samples from the fish, do you have to kill the fish to take the sample, or would it be enough small milligrams or biopsy of flesh of the fish without killing it?
So it depends on the fish and the age and a couple other things. Some fish are much more resilient to taking biopsies and will be fine. Some fish we can only get as very, very small juveniles. And so we sacrifice them up front using humane practices to make sure that we're not biopsying. I think, but especially if you think about shark fin, right? For example, very easy to work with a reliable partner and get a fin clip from a shark when they're doing tagging as regular operation in the ocean, and we don't have to then harvest a shark for our own purposes, which would be kind of a very difficult thing for us to stomach.
And I guess as more it also develops these culturing techniques, as less tissue you need actually to develop a new culture. So definitely that's
Yeah,
Now,
the learning definitely accumulates, right? So from one species to the next
yeah,
species.
especially if you want to harvest from species where individual specimens, where you know they have different growth rates. It's like Mendelssohn, like genetic selection, basically, almost, without doing
Yes.
genetics, but just straight growing them in tissue culture. Straightforward. Another question for anonymous attendee: how difficult is it to ensure batch-to-batch consistency for cultivated products?
I think we don't know yet 100% because we haven't gone to production scale, right? So this is a bit of a question that we'll find out more. But I would say, in general, the biggest challenge is going to be consistency of the food and ingredients that are getting put into the process, because there's some variability from your cells, but all of your cells are basically frozen down from a stock that is started from one clone. So those should be mostly well controlled. And historically, those have been quite well controlled. But when you have sugar that is more or less pure than your spec, for example, that's where you introduce kind of some variations that are harder to control. But machine learning and AI is getting incredibly good at process control. And that potentially is a step change for managing variations that could occur.
But I suppose basically the medium, where you add your nutrition, your culture medium, your growth, well, not growth hormones, but nutrition levels you add to the cells. And of course, if you want to make things cheaper, you might also change your cocktail of ingredients over time. And this might indirectly affect then the cell growth. So that's something which probably has to be monitored and modulated constantly. It's not a once-off development.
No, you have to build your own quality standards internally. And we're having to do this with our suppliers now and basically qualify the whole supply chain. Because actually many providers are food grade, but food grade is actually a broad range of things. And so you have to define for your supplier what you actually care about and what can vary a little bit.
So amino acid composition, for example, or sugars, carbohydrates. And
or even water content. I mean, all sorts of things that when you then reconstitute media can lead to differences from what you expect to have.
And this is not only for cultivated, I guess for plant-based food, also very much the same. There's another question from Anand, who's very curious. Do these cultivated fish get infected based on temperature and storage? If yes, what microbes are these?
So not so much based on temperature and storage. I mean, the microbes have to come from somewhere, right? So that's always a starting point. And we're growing them in what should be a sterile bioreactor environment. So it's a steel container that's been steam sterilized or potentially cleaned in place using a traditional kind of process. So typically the contaminants come from room air, and they're just adventitious contaminants. So you'll get potentially E. coli, you could get potentially all sorts of other things that happen to be around in a food manufacturing facility in the air, and normally are not problematic to anybody. But if they get into an environment full of highly nutritious feedstock, they double much faster than cells, so they can overtake culture.
So I guess the question is also using antibiotica or something like this to reduce the risk, or not?
No, it's process control, right? Because if something gets into your process, antibiotics at the level you would want to use them to control an infection would not be something you'd want to feed people. So in an ideal scenario, it's sterile going in and it's clean all the way through. And that's how we're doing our scale up. The only antibiotics that are used typically are in the early harvest process, because there's a high risk of contaminants from the seawater or whatever that may get into the tissue that you're harvesting. But usually after 3 or 4 days, those get removed and we're running antibiotic free.
And the same, I guess, is also for plant-based food. It doesn't really matter what you grow in tissue culture. All of them could be potentially prone to infections or microbes. I
think plant-based has a different set of risks, right? Because you have aflatoxins and other things that come from the actual food staple and not from the production process so much.
And have to be also considered and monitored, right?
Right.
I think we run out of time and questions. Thanks very much for really comprehensive answers to all of these very diverse questions. And I'd like to close up. Thank you again, Mihir, Wasamon, and also Thimo, for talking more about food innovations and potential health aspect. And I think all the reviewers, viewers, and questionnaires really nicely answered, and maybe opened really up your views to also being part of these new developments of foods for the future. I thank you all very much and enjoy your evening. Thank you.
Thank you.
Go on.
Bye-bye.
Cheers.